In a completely randomized experimental design, basically what we do is we put all the subjects' names in a hat and we just draw names out at random and assign them into treatment groups. The type of experiment you've been doing thus far is called a randomized comparative experiment, and that's where we have 2 or more treatments and we randomly assign people into those treatment groups. Towards the end of the chapter, we'll look at 2 other types of experimental designs, a block design and a matched pairs design. For all of these designs, we usually have something called a control group, and the control group gets a standard treatment, or you can think of it as being like the status quo. If this was a medication that we were testing, one group would get the actual medication, and the control group would get a placebo, which would be a sugar pill that looks like the medication but doesn't have any medication in it. And that allows us to compare and see whether the medication or the treatment is actually helping. Okay. Kind of the logic or reasoning behind why we have these randomized things is that when we randomly assign our subjects into treatment groups, this means that all the groups should be fairly similar, except for the treatment that we're applying to them. So it's very similar to the idea of a random sample. If we've taken a random sample, it should in generally look like our population. So when we randomly assign our subjects into treatment groups, the groups are fairly similar. And what that does is then any outside influences that we haven't accounted for, any lurking variables or things like that, are going to act on all the groups similarly. And so we don't have to worry about that creating a whole lot of bias for us. And then because we randomly assign, any difference that we see in our response variable is due to one of 2 things. So either it's the actual treatment is working or doing something, or there's some sort of random chance going on that's kind of tweaking our results. And so this is why we generally replicate our experiments. That way we can ascertain whether it's the treatment that's causing the difference we see, or if it just happened to be that time that there was some random chance at play that caused us to see a difference. Okay, so these ideas are going to form the 3 principles of experimental design. So come back in the next video and we'll take a look at those and talk about them in more detail.